Okay. So now we're in chapter 12. This is part A of 4 parts, but we talk about the central nervous system here. And in particular, what we're going to try to discuss is some of the brain functioning, right? So we'll talk about that and get into some of these parts and areas that we talk about in the brain. But first, we'll discuss kind of an overview of some of that, including some of the fetal development stuff, right, in chapter 12. Okay, so we know that the CNS is the brain and spinal cord. That's obvious already, so you already know this, right? So we first encounter our first word here, or first term here in the CNS we call cephalization, right? Cephalization is the formation of the brain. And we're talking about this in fetal development, so we got to go back to fetus developing. And in fact, we take a look at evolution because the reason why we take a look at evolution is because the human brain is the most highly advanced and biggest brain out there in the land of, animals, if you will, right? Got the biggest, most developed one. That's changed over time because our brains are actually getting bigger and bigger. Our heads are getting bigger and bigger because we're holding more brain tissue or neurons, right? And in fact, the place that we've developed more of the brain part, that we've developed more of the brain than anything else over time, is the rostral or the anterior portion of the brain. That's that big old frontal part of the brain. And we'll see why that one's important because we'll talk about the cerebrum, right? Cerebrum is what we're talking about. So we start off in the, even before we sit the fetus, in the embryo, right? And we talk about the first formation of this tissue being the neural tube. It's a developmental milestone for new life, right? Is to see the first formation of the neural tube, and that's an indicator of, somewhat of a normal development, because that's going to actually become the brain and the spinal cord, right? And so when the neural tube begins to form and then begins to expand and kind of fold itself in, we have these 4 major areas, right? We call these vesicles, right? But these 4 areas: the forebrain, the midbrain, and the hindbrain, right? The forward brain, the middle part of the brain, and the back of the brain, right? We call this the prosencephalon, the mesencephalon, or the rhobencephalon. And these are going to become then these parts of the brain that we're going to see later on, okay? Which is part of the front of the brain, becomes the cerebral hemisphere. And actually, we have 2 hemispheres, right and the left, but that becomes the cerebrum, right? When we talk about cerebral hemispheres, it's the cerebrum. And the diencephalon becomes the epithalamus, the thalamus, hypothalamus, and part of the retina, right? Actually, it's the outpouching of the brain, the back of our eye, in fact. Mesencephalon becomes the midbrain. The metencephalon becomes the pons and cerebellum. The diencephalon becomes what we call the medulla oblongata. And we're going to see this cavity in the neural tube then become what we call ventricles, which we'll see also in a second. Figure 12.1 shows you the neural tube. It's really early in life when that occurs. The primary brain vesicle, where you see the 3 main areas: forebrain, midbrain, and hindbrain. And then how it sections off, right? So we see that secondary brain vesicles. And then finally, part D shows you what we have, right? As far as development, where those come from. That's all we're looking at, where they come from. And in fact, if you look at this stage of brain development, part C, and you match that up with about the same time frame as other animals, kind of looks the same. And in fact, they all resemble each other at that point. And obviously, they're going to develop differently. So there's there's images out there that you can look up that show you development, brain development of let's say a fish and a bird and whatever else, and a human, and they all kind of look the same in part C. Really interesting stuff. So the brain grows faster than the membrane skull, right? The connective tissue, if you will, the cartilage and what's now becoming bone, the fontanelles, right? All that. So what occurs is that since the brain is growing in a very fast pace, it's going to start to fold in itself. It's going to start to fold in and fold out. And so when it does that, we start to see this folding, if you will, all throughout the brain. The forebrain is going to go towards the brainstem. The cerebral hemispheres go double back, so they go back and forth. So in fact, you can see week 5 here. This is very early on. And you can see this folding, if you will, starting to occur back and forth. Then you start to see then the brain itself forming. And that's where we have these folds, if you will, in the brain tissue. And that's what we're going to, we'll see this week when, after our exam, we'll look at brain, right? We'll look at the brain tissue. All right, so on to adults, right? Adults have 4 regions, right? We'll visit or try to visit each one of these. And in particular, we want to really concentrate on the cerebral hemisphere because I think that's important. That's the one area, major area, that actually sets us apart from other animals. Because we have a huge cerebral hemisphere, highly developed. Because of that, that's where our thoughts are, that's where our processing is of those thoughts, that's where emotions come in, parts of those places. That's where reasoning comes in, language, those type of things, right? We'll try to visit the diencephalon, the brainstem, and the cerebellum, although we won't talk a whole lot on cerebellum. Something we're just going to have to kind of cut away at, right? But here's our at birth brain, right? Here's our folds that we're talking about because we're running out of room. So what we've done here is kind of folded the brain on itself, and you'll notice that a large part of the brain is the cerebral hemisphere or the cerebrum. The other areas like the diencephalon, the brainstem, and even parts of the cerebellum are actually ancient, old parts of our brain. For example, some of those hold emotions like rage and anger, right? Because those are emotional, those are features that we've had for a very long time. The cerebral hemisphere is new. And what I mean by new is it's new in our evolution. It's the newest feature. And that's the part that's, evolutionarily speaking, is expanding. It's getting bigger and bigger. Okay, so we'll take a look at the regions here. Of the brain. So when we talk about gray matter and white matter, again, we're talking about myelin, right? With myelin, without myelin. The gray matter in the brain, for example, is the short, unmyelinated neurons, or those neurons that have myelin but just the cell bodies. Those are considered gray matter. And white matter is the myelin and non-myelinated axons. So here, you do have a pattern in the CNS, right? Not only in the brain, but in the spinal cord, where you're going to have a central cavity surrounded by gray matter with white matter and then gray matter again. That's kind of how it's put together, even though there's different, amounts of each one. This is what we see. So here we just took a section of the spinal cord and looked at it, cross-section view, and see then there's a central cavity, gray matter, white matter, and then at the very edge, gray matter again. So it's following that pattern. But as you can see, as we go up further, we see it changes slightly. You see the central canal. This is in the brainstem. The central canal, cavity's bigger, gray matter, white matter, and then dotted gray matter again. So it changes slightly, but it's almost the same. It's the basic pattern as far as that's concerned. Look at the brain, right? There's a central cavity, and in fact, there's some other cavities there. Gray matter is now like scattered around, but gray matter there. White matter, and then there's a line of gray on the very edge. Pattern looks different, but nonetheless, almost the same pattern in the fact that what comes first, second, third, fourth. Okay, ventricles, right? Ventricles are these cavities or these chambers that are filled with fluid. They all connect, so that's not like they're separate, they all connect. Even in the spinal cord, they connect to the cavity that's in the middle. We call that a central cavity. And they're all filled with an extracellular fluid we call cerebrospinal fluid. All of those areas are lined with ependymal cells, which is a glial cell, right? So the biggest ones we have are located in the brain, and they're deep inside the cerebrum, and it's a pair. They're C-shaped, and we'll call those lateral ventricles. Again, they're a pair. There's one on the left and one on the right. The pair is separated by this membrane we call the septum pellucidum. Those pairs are both connected to one chamber we call the third ventricle. And we've got a connecting chamber between them. It's called the interventricular foramen, right? It connects the lateral ventricles to the third ventricle. The third ventricle is in the diencephalon. The third ventricle is finally connected to a fourth ventricle through a channel we call the cerebral aqueduct. The fourth ventricle is in the hindbrain, and that fourth ventricle is then connected to the central canal. Okay? So again, lateral vesicles, ventricle, third ventricle, fourth ventricle, then central canal. And you might ask yourself, where's number one and number two? Well, lateral are a pair, And we don't name them number 1 and number 2 because there's no indication where one is the right one or the left one, but it's the pair, 1 and 2. Does that make sense? Because that was never explained to me when I took A&P. It's just like there's lateral ventricles. But that's why. It's 1 and 2. 3, 4, and then central canal. Okay?